Friends, welcome to Biology Made Easy. Today in this video, we'll discuss the mode of enzyme action. Already you know what is the main role of enzymes. Their main role is they will convert the substrates into products. But how? Usually, each enzyme has some active sites. And to this active site, the substrates combine. The substrate combine to the active sites and they form one enzyme substrate complex. Then this enzyme substrate complex is converted to products and enzyme is set free because enzyme remains unaltered at the end of the reaction. So this is the basic idea how the enzymes function during its action. It has some active site. The active site they function like a key, while this acts as the lock. As a specific key fits to a specific lock, similarly a specific enzyme acts with a particular substrate. This is a substrate and this is enzyme. The enzyme function like key, while the substrate function like lock. But how it operates? Whenever any reaction takes place, the different molecules they randomly combine with each other to give rise to different types of products. So when we'll take some chemical reaction inside the cell, it can take place in absence of a catalyst, or you can tell biocatalyst like enzymes, or it may function in presence or absence of enzymes. If enzymes are completely absent, then it requires a lot of external energy to activate the molecules so that there will be random collision. That is known as activation energy. That is activation energy. That means without enzymes or without catalyst, the activation energy is very high. But if you give catalyst, in case of inorganic, will give inorganic catalyst, and in case of cells, will give organic catalyst, that enzyme, then the activation energy will be lowered. If we take one example, suppose let us take the example of carbon dioxide and water, and it will be converted to bicarbonic acid in presence of enzyme carbonic anhydrase. If there is no enzyme, if this carbonic anhydrase is not there, then it will give rise to 200 molecules of this bicarbonate per hour. That means per hour, only 200 molecules of bicarbonic acid will form because the reaction is very slow. Now, this same reaction, carbon dioxide plus water in presence of enzyme, with enzyme, then it will give rise to 6 lakhs per second. 6 lakhs per second with enzyme, while only 200 per hour without enzyme. This is the basic difference. That means when enzyme is there, the rate of the reaction is increasing a million of times. Million of times. That is the difference between enzymes and without enzymes. With catalyst, without catalyst. So the main function of the enzymes is to lower this activation energy so that the reactions can take place within normal temperature. Let me give you another example. Suppose you will be asked to liberate oxygen. So what you will do? You take potassium dichromate and you just heat it. Oxygen will liberate it. But at what temperature? 1000 degrees centigrade. Means you have to go to a blast furnace. But if you will be asked to liberate oxygen in the laboratory, you can do it. To potassium dichromate, you add some catalyst. Then in 100 degrees centigrade by Bunsen burner, you will see that oxygen is liberated. So from 1000 degrees centigrade, without any inorganic catalyst, it was liberated. But when you are giving catalyst, potassium dichromate with the manganese dioxide, then it is liberated 100 degrees centigrade. But you see, this same oxygen is liberated in the green cells of the plants at what temperature? Normal temperature, 15, 20, 25, 30, like that. How is it possible? It is possible because inside the cell there are biocatalysts known as enzymes. Again, this is a difference between inorganic catalyst and organic catalyst. Inorganic catalyst, their efficiency is less, though they can function in high temperature. But the organic catalyst, the enzymes, they are more efficient and they can function at a low temperature. The same oxygen which are requiring 1000 degrees centigrade without enzymes, without catalyst, now with inorganic catalyst 100 degrees centigrade, and with organic catalyst 10, 20, 30 temperature. This is the basic difference. So, how the whole things happen? When the substrate combines, when the enzyme at the active site of the enzyme, when the substrate combines, it gives rise to enzyme substrate complex. Inside this enzyme substrate complex, there will be certain bond breaking, making, and the shape will change. Then it will give rise to products. So here you see this graph. This graph is very important. So here you see this is the substrate. In the x-axis there is the progression of reaction. In y-axis there is the potential energy. This is the substrate. This is the product. So if there is no enzyme, without enzyme, you see this red line. This is the red line. This is the substrate. This is the product, and this is the rate of this is the activation energy without enzyme. But when you give enzyme, you see this green line. Now this is green line. The same product is produced from the substrate, but activation energy is low. That means when we are giving enzymes, the activation is lowered. But here we have to understand how this reaction is taking place. Is it taking place in a single step? Just the enzyme substrate will combine together and immediately the products will be released? No. Because in between there will be several transitional steps. The enzyme will combine the substrate. There will be bond breaking and making to give rise to another ES complex, enzyme substrate complex. Again there will be bond breaking and making to give rise to another substrate. Then again there will be bond breaking making to give rise to another substrate. That means from this point to this point, the total range is known as transition state. This is known as transition. This is known as a transition state from this to this. That means this reaction doesn't take place in a single step. There are so many steps. There will be combined together, bond breaking, making with a change in the shape of the substrate. Then it will give rise to another enzyme substrate complex. Then another. Let me give you one analogy so that it will be very clear to you. The smartphones you are using, here you will get different types of steel materials are also used. Your laptop, your computer, different types of steel, fine steels are there. With that steel, the rockets are prepared. Again, with the different types of irons, the different vehicles, train, bus, car, motorcycle, all are synthesized. With the same iron, the needles are synthesized. And all these steel, irons, even the railway lines, also iron. That means all the type of steels, irons, they are ultimately coming from the iron ore, which we are getting from the mines. So do you think that in the blast furnace we will give this iron ores in one side, and in the other side the smartphones, laptops, computers, all these things will be coming out at a single step? Naturally the answer is no. Because the iron ores, they will melt in the blast furnace, then it will be converted to different types of iron with some impurity, then to the impurities some again chemicals will be added, that will be purified, then we'll get different types of irons, then to that again iron, again some chemicals will be added, it will convert to steel, again the steel, some there will be certain things, then there will be fine, and then we are getting all this. That means from the iron ore, when we are getting the fine steels, there are so many transitional steps. If our aim is to construct a mobile phone or some laptop, that means we have to go through several transition states, and all these transition states are unstable, they are temporary. The final product is stable. Now you apply that analogy to this point. When this enzyme and substrate combine together, there is bond breaking and making, and it will give rise to another enzyme substrate complex that is unstable, that is temporary. Again there will be bond breaking making, give rise to another enzyme substrate complex that is again unstable, temporary. Again there will be bond breaking making, and again unstable and temporary. So there is several transitional states, several altered transition states and each state is unstable temporary. It will be permanent, it will be stable only when we are getting products. So in between there are so many transition steps and that this is the range of that transition state. So I hope you have understood it perfectly from the analogy also. So when substrate and enzyme combine together, there are so many transition states and finally the product is produced. P, I have written that P. Now again you mark the energy, this is the substrate, this is the product. That means this is the energy level of the substrate, this is the energy level of the product. That means the product has a lower energy level than that of the substrate. So when a product has a lower level, that means liberating energy, that means such type of reactions you can call exothermic reactions. Exothermic reaction. Because it liberates energy, its energy level is low. So what happens to remaining energy? That has liberated. Suppose it would have been somewhere here. Suppose it would have been somewhere here, above substrate level. That means it requires more energy. That means in that case the product energy is less than substrate energy. That means that is the endothermic reaction. Endothermic reaction. So how the exothermic and endothermic reaction take place and what is the mechanism, what are the different transition states, this is very, very important. And if you have understood it perfectly, then you can answer all the questions. So now if we will summarize how these things are taking place, then we can come to the conclusion. What is happening? First point, enzyme combines with the substrate. Second point, there is a bond breaking and making. Bond breaking and making. Then shape of substrate changes. Again bond breaking and making. And again there will be change in shape. And finally you will get the products are formed. Products are released and enzyme set free. Enzyme set free. That means the enzyme will combine with substrate, and in the enzyme substrate complex there will be bond breaking making, and again another enzyme substrate complex formed. Again there is bond breaking making. So these are all transition states. And when the product is formed, the products are released and the enzyme is set free. This is the mode of enzyme action. And for this type of reaction, usually two theories are there. One theory is known as lock and key theory. Lock and key theory put forth by Emil Fischer. So according to him, the substrate functions like a lock and the enzyme functions like a key. But this is not acceptable at present because this structure of the substrate should fit to the active site. But later it was replaced by induced fit theory. Induced fit theory proposed by Osland. And according to him, when enzyme and substrate combine together, the shape of the enzyme changes, and he has observed it through electron microscope. That means the structure of the enzyme before enzyme substrate complex formation and during enzyme substrate complex changes. He has observed it through electron microscope. So this is most accepted, and in that way also we have discovered the structure changes. Now if we will go for questions. One question you can write: the transition states of enzyme substrate are unstable, stable, C, no change, D, none of the above. This is one question. Similarly, the second question, the graph just I have drawn, that graph will be asked. The graph will be given like this. Then here it will be written, suppose this is A, this is B, and here it will be written C, here it will be written D, and you will be asked what is A, what is B, what is C, what is D. So from the graph you see this is the activation energy without energy, this is the activation energy with enzyme, this is C is substrate, the D is product. Again you can be asked what is there in the x-axis and what is in the y-axis. So in the y-axis there is a potential energy, and the x there is the progression of the reaction that we have drawn in the graph. So simply we remember this graph, and whatever this question was even asked once. So you remember the graph and you will be able. So these are the type of questions that come from this mechanism action. So thank you very much, and have a good day.